please.
Thank you, Josephine. So first, I'd like to thank Martin and Lars who have given me the opportunity to present to you the work we're doing over in my team at Stockholm University. They gave me quite a difficult task to be able to present and start this wonderful seminar about lignin. And as Josephine said, I've entitled my talk Genetic and Cellular Regulations of Lignin Composition in Herbaceous and Woody Plants. So let's directly, I hope you all see my presentation. Okay, I hope so. And let's go directly in terms of what is this essential molecules we want to work on, lignin. It's insoluble and a very abundant plant aromatic polymer. It's actually the second most abundant carbon biopolymer on Earth. And it derives from two aromatic amino acids common to all living organisms, tyrosine and phenylalanine mostly. And these are modified through a set of enzymatic steps in the plants into typical phenylpropane structures, which have an aromatic ring, which is differently substituted, as well as an aliphatic tail with different functions, as you can see here. And these monomers of lignin are going to be secreted in the wall and assembled in a very complex polymer in different ways, thereby making lignin as we know it, which accounts for about 25 to 30% of plant dry mass. And this varies with the organs considered, with the developmental condition, with the species, and with many things. In terms of plant development, lignin is essential, as if we genetically reverse engineer lignin to reduce it from its native 100% to just leave 5% residual by stacking genetic loss of function mutation, what we get are two plants of the same age. And you can see we have the two plants here. That's a zoom of the plant. And you can see how much the loss of lignin affects the entire development of the plant. And lignin has thus been associated to multiple functions in plants, such as diffusion barrier or waterproofing, resistance to biotic degradation, both herbivores and pathogens, mechanically strengthening plant organs to sustain different gravity vector or to be able to grow straight when they are on inclinations of a hill, or even in the hydromineral sap conduction. So this lignin, comes from the basic biosynthesis of that polymer directly in the wall. And from a classical biochemical point of view, I'll try to simplify it to be able to show you what models we're working on. So here's an artist's rendering of a plant cell, with in white here the cytoplasm of the cell, which is delimited by its plasma membrane. And outside here in brown, we have the cell wall. So these lignin monomers are going to be made in the cytoplasm, and then they're going to be secreted into the cell wall through the plasma membrane by mechanisms we don't really understand at the moment. These lignin monomers are then going to be activated in the cell wall by specific enzymes. We call them phenol oxidases. And these phenol oxidases can use co-substrates to make a phenoxy radical. Based on the conjugated insaturated structures of the lignin monomers, the charge is going to resonate, and alter, and multiple monomers are going to resonate together. And when two radicals are going to meet together, even if they are in different resonance form, they will cross-couple to be able to make a first dimer. Then, after subsequent activation of the dimer into a phenoxy radical, either directly with the interaction of the phenol oxidase or via mediator molecules to be able to displace this radical charge, are going to enable the N-wise extension of the polymer in order to lead to the entire lignin structure. And here I wanted to show you that each lignin monomer is differently linked. As you can see, you've got linkage, which can be, as we say, beta O4, beta 5s, beta beta, or 5-5. And I showed basically the differential enthalpy for each one of these bonding. Here, I have to say this is a simplified representation where we consider that we only have this compound as the lignin monomer. However, in the resulting polymer, residues are going to have specific position. So we're going to have some residues which are internal here in black. Other residues are going to be N function with a free aliphatic. Others are going to be N functions with a free phenolic ring. And some are even going to have both. This is quite important for the branching and the reactivity of the lignin molecule. So this is quite nice, but what is exactly the lignin monomer and how does it vary in terms of composition? I showed you that the consensual belief structure is this phenylpropane unit with an aliphatic tail, which can vary in terminal function. It can be an ester, a carboxylic acid, an aldehyde, or an alcohol. But also that the aromatic ring itself can be differently substituted, mostly in meta and para position, with either hydrogen, a hydroxyl, or methoxyl group. These are being, until a couple of years, as the main lignin monomers. But that's not exactly true. In plants, what we've gotten, depending on the cell type and the species, is much other types of monomers diversity. And they can include coumaroyl esters, they can have these classical cinnamyl alcohols that we've talked, but also benzaldehyde, stilbenes, flavonoid, all which have in common the fact that they have a conjugated unsaturated structures with an hydroxyl capable of making a phenoxy radical. And then after having all these cross-coupling that we've talked about. So, I mean, the lignin monomer is at the moment, it's a bit difficult to define what it is. And even these molecules that I showed, we're not even sure if they are themselves the monomers or just an intermediate step to be able to make the truly lignin monomer, the one that will be secreted in the wall and polymerized to form lignin. So this lignin is, compared to the other cell wall component, a relatively young molecule. It appeared 450 million years ago when plants decided to shift from a water-based environment to a land-based environment. And they needed to be able to do this transition because the air is dry. And basically air is also 1,000-fold less dense than water. Both vasculoskeletal systems, so both a skeleton and a vascular system, needed to be established. And these coincided with the appearance of vascular plant, which required the appearance of lignin. And this is a bit reminiscent of the first image I showed you when we genetically engineer lignin out of the plant. It could not stand, it could not grow. So it really requires this lignin to be able to grow and develop. So from this evolutionary point of view, I've just wanted to do a very simplified profile between the early types of unicellular algae up to the first land plant mosses, which don't have a vascular system, and after the vascular plants, okay, from the most primitive lycophytes up to flowering plants. So if we characterize the different compounds, although it's kind of complex for the unicellular algae, and it's still debated whether or not they've got a couple of phenolics, we can see that most of the species have got phenylpropanoids, or at least the capacity to metabolize them, as well as flavonoids. But the presence of lignin is independent from the presence of these potential monomers. All the kind of unicellular algae, as well as the first moss land plants, do not have lignin. And lignin specifically appears with all the vascular plants. This lignin, moreover, diversifies between the vascular plant, between the land plants that we see all the time, from the different units composing their lignin. And there I just want to highlight and remind you that the aromatic ring could be differently substituted, either with one hydroxyl or methoxyl, or two methoxyl and one hydroxyl. making different types of units that we classically call H for hydroxyphenyl, G for guaiacyl, and S for syringel. And if we analyze the lignin composition across these different species, which go from primitive lycophytes to hardwood species to grasses to hardwood species, we see that they share some composition and some diversity, but some, such as some conifers, have lost one type of lignin monomers. So this is quite interesting, and now with the extensive amount of data that we have on the genomes, we can actually retrace and reanalyze which genes are present in which species, encoding for these different enzymes which will enable these different monomers to be produced. So here again, I go back to my rough evolutionary tree, and I'll show you here the number of gene coding proteins, okay, which for sure in the unicellular algae are less than in the land plant. But you see, between the Arabidopsis kind of evolved vascular land plant and the Fiscomitrella, which is just a moss with no lignin, we have about the same numbers of gene coding transcripts. So we wanted to retrace the specific enzymatic modification of the aromatic amino acids into the different phenylpropanoids I've just showed before. So we'll take genes after genes and compare whether or not they're present and in how many copies they're present in these different organisms. So when we look at the first gene encoding for the hydroxylation in PAL, what we're seeing is that it's exclusive to land plants, independently of the presence of lignin or not. Then what we've got is a second type of hydroxylation to make these kind of catechol structures, which are also present in all land plants, independently if they have lignin or they don't, like in the moss. And the third types of hydroxylation here, making these 5-hydroxy guaiacyl substitution, you see it's much more specific to evolved vascular plants, such as showing that Selaginella as a lycophyte doesn't have it. This is actually in case of the true F5H of the angiosperm, Selaginella does not have a similar gene, but it's got a different gene which also catalyzes the same type of conversions. If we look at the second aspect of lignin monomers, which is the change of the aliphatic tail, we can see that the CCR here, which allows to reduce carboxylic acids to basically aldehyde, it's a bit more in terms of co-thioesters, we see that they're present in all species, even the unicellular algae. And if we take the step which is after, enabling the reduction of the aldehydes in terms of alcohol, we see that they're even more abundant and present in all the different plants. So it's kind of very difficult to pinpoint what is the biosynthetic process enabling the production of specific units enabling lignin. And this has evolved sequentially during evolution to be able to gain either in a similar way or differently similar units. So all of this, lignin, is only important when we consider the unit of any living organisms. And in plants, the unit, like us, is the cell. So how is the relationship between the lignin composition and structures and the cell itself? So here I just wanted to show you a cross-section of one of my favorite plants. It's Arabidopsis thaliana. I showed you already a couple of images of this. And in white, you have the autofluorescent properties of the lignin. So if we zoom up in one of these structures, which is highly containing lignin, we can see that it's made of different cells just by the morphology, and they have different intensity of the autofluorescence, which is more or less proportional to changes in composition and in amount. So when we look at the different cells, these different cells are made to fulfill different functions, and we can see within them the protoxylem vessels, which are playing a role in the early vascular conduction, the metaxylem vessels, which are playing a role in terms of the continuous vascular transport and hydration of all the tissues. We have the auxiliary fibers, which are made as a strengthening element for the plant against mechanical stress. We have the intervascular fibers, which are different types of fibers with thicker walls, which are also associated to mechanical resistance. And lignified parenchyma, which are enforcing the cohesion between the cortical areas and the medulla. So this is quite nice. And if we go towards some rough biochemical analysis, what we could see is that these different cell types have actually different amount of lignin and different composition of lignin, which is between cell types, but also between the cell wall layers of each cell types. And here I just wanted to do a rough artist rendering of the same vascular structures we just looked with this bundle here and the intervascular fibers and lignified parenchyma on the side, and color-coded these hydroxyl unit, these hydroxyphenyl units in red, the guaiacyl unit in blue, and the syringyl unit in green. Okay, so this is an absolute, and every single cell is much more of a ratio. Okay, so they include a bit of all the monomers in different proportion. But you can see here that the protoxylem vessels, what they have is low amount of lignin, which is enriched in guaiacyl unit. The metaxylem vessel have got high levels of lignin enriched in guaiacyl units. But the auxiliary fibers, which are just next to the metaxylem fibers, vessels, have got high level of lignin, which are enriched in S units. If we go a bit further in terms of the intervascular fibers, they've got medium amount of lignin, which is enriched in S units, whereas the lignified parenchyma have got lower level of lignin, enriched in S units. Here. Additionally, what you can see is that in between the layers of the cell walls, that the layer in between the cells is enriched in H units compared to the much more secondary cell wall layers. Okay, so what we're having is a great diversity of the different lignin in between the cells, and this is actually change between species of vascular plants just because we have a different proportion of these different cell types, which are assembled differently. So, for example, here we see Arabidopsis thaliana, okay, which is a weed, okay, it just grows within 1 year, but you've got the same cell types organized differently in poplar wood, or even in grasses like Brachypodium, which is like a cereal grass. Good. So what we are having is actually different proportions of different cell types depending on the species. When we look at the cell itself, lignin, as I said, is differently deposited in different places of the cell wall. And here this is one isolated lignified sap conducting vessels. And you can see that in blue, we've highlighted the autofluorescent properties of that lignin, which is restricted to the secondary cell walls. If we look at this cell and section it to go in terms of electron microscopy, we see similarly that these ribs, these are the ribs that we're seeing here, are also darkly stained with potassium permanganate, whereas the primary cell wall is not stained, or not autofluorescent portion here is not stained. Really showing that lignin itself is not just diffusing everywhere, it is spatially restricted to defined site in the cells. If we go a bit further in terms of the cell wall layers in cells, because the cell wall is assembled by multiple layers, we can see different organization of the lignin, going from the primary cell wall, which in this case for the vessel doesn't have lignin, to the primary cell wall to S1 layer, where we have organization of lignin, which is much more spherical. And if we go to the S2 to S3 layers, the one which are closer to the lumen, we're getting much more rod organization of the lignin. And this has been reported for multiple species as a gradient from the cortical to the medulla part of the cell wall of each of the lignified cells. So lastly, to be able to put lignin in terms of its context, it plays, and we've seen it's different from every specific cell type. So it's established and deposited within cell types in specific timelines. And we can also film, and we've done this using inducible cell cultures, to be able to film when lignin appeared in plant cells. So we have got a small little movie where you have one cell here, which is going to put on its secondary cell wall, specifically lignin, and it's going to appear at one specific time during its development. And then you can see here that it's going to increase and change composition during time, well, change quantity. And here's just the cell with one frame every 10 minutes, and you're seeing very quickly, I'll try to put it again and stop. Up here you're seeing the depositions of the lignin specifically in the cell, which increase with the time. Let's look at this again. Good. Just illustrating that lignin also appears at specific times in specific cells to mediate their function. So there's a developmental regulation. Lastly, lignin is also responsive to environmental constraints and dependent on environmental condition, such as the inclination of the soil, the proportions of the wind, heat, humidity, illumination, any kind of climate change condition. This will affect the development of the plant, of the cells, and thereby of their lignin, specifically in each cell type. Nutrient availability is also going to modulate this, either increase or decrease or even change the composition, as well as the presence of pathogens and herbivores. In each cases, every single cell will integrate for its lignin amount and composition and modulate its organization, its cell wall layer deposition, and in which cell type it's going to be placed. So we have something extremely dynamic. So our current model, which is essentially based from bulk analysis of organs and in vitro synthesis, assembled this complexity resulted from a random assembly, which was basically controlled by a monomer availability. And yes, for sure, you can now understand that each time we take a plant organ, we're basically averaging cell types, developmental time, integration of the environment, and subcellular regulation. So there's some kind of misconceptions of how lignin is regulated due to an averaging effect, averaging different lignins in different conditions, differently responding to the environment in different cells. And at the moment, we are a bit stuck in between, is this the monomer? Is this how the lignin is? Is it assembled in a specific way? And there's no actually clear link between specific lignin in specific cell types or during their development or in response to climate change and their function at the cellular level. So in order to go about this, what we decided to do is to be able to reconcile these conflicts, and these are not really conflicts, but these different scale conceptions between the biochemical models that I presented, the cell biological model, the molecular biology with these multiple genes, as well as the developmental model, to get an integrated understanding of lignin, where we could consider how the diversity in the monomers, composition, amount, and position could be regulated, how this could be mediated by a genetic program which was acquired during evolution and even lost in specific species, like we've seen for the hardwoods, that it could be integrated for the proper development and integration of environmental constraints, and as well be cell-specific, because each cell fulfills a specific role. So this integrated approach, what we wanted to do is to be able to re-examine lignin, but with a focus at the cellular and subcellular level, directly in whole tissues and organs. Okay? So be able to minimize this averaging effect of taking basically an entire tree or an entire tree trunk and grinding it to be able to get the lignin. So in order to go about to do this, we've got a couple of experimental systems. One of the one that we use is a purely in vitro biomimetic process. And this is the formation of synthetic lignin, completely artificial, okay, in the tube completely in vitro, okay, so no biological context. And these are called dehydrogenation polymer, where we can take our favorite monomer and be able to make some type of artificial lignin. And this is yet again a rendering. I mean, I don't know if that's the true structures, where you've got a lignin molecule made of coniferol alcohol, different interlinkage between the units, with one unit with a terminal end and a free aliphatic, and one with a free phenoxy end. So we can make these synthetic lignin using different processes, where we can take the different substrates making lignin, the phenylpropanoid, and in that case, hydrogen peroxide. We take one of the phenyl oxidase, peroxidase, and we can mix them together to create in a bulk these different monomers. We can also try to mimic the kind of supply which is controlled by biology, as I showed you, in a kind of artificial way, by gradually supplying the monomers and the hydrogen peroxide to the enzyme. And here we can see and we can modulate how the polymer is going to assemble. And this can then after be monitored using size exclusion chromatography, where we can see the difference between our polymer. This is just our calibrators in or molecular weight, compared basically to our solvent itself. So what we're getting is a larger polymer. The second experimental system that we have is purely biological. And what we're using in my team is two complementary systems. The first one is Arabidopsis thaliana. And for these Arabidopsis thaliana, we have a collections of loss of function mutant in which we removed one, two, or even multiple genes encoding for enzymes which do specific modification, the same enzyme as I showed you initially, which were acquired during evolution. And you can see each time that these are cross-sections of the stems, equivalent parts, and you can see that what we've got is different organization. And this enables us to restrict the lignin composition by removing certain capacity. So for example, in the FAR1 mutant here, what we do is we take a hardwood type of lignin in the typical angiosperms, and we remove all the S units genetically. So it's actually like softwood. And we can do this and test how this is affecting the different cell types. This system is great because we can actually engineer the lignin composition as we want, I mean, to a certain degree. But it doesn't have all the developmental context, okay, these sequential development of lignin, as I showed you in the short movie. So for this, we associate this to a second system, which is hybrid poplar. And in hybrid poplar, what we can use also is RNA silencing to be able to reduce specific genes. And when we look through the wood development here, and this is a section through the wood where you can see different cross sections of the different cells, a bit like what I showed you before. What we actually have is a timeline of the development of the different cell types. Exactly like what you can see in dendrochronology, is the further you go within the center of the section, the older the cells you're going to get. And we can actually isolate RNA and the gene expression in these different sections, and we can monitor the expression. And here you see 3 of the genes that I previously showed with encoding for enzymes enabling the first, second, and third hydroxylation of the aromatic ring. And you can see how they are expressed differently across the wood forming organs. So this is quite nice in terms of this, but we still can't follow exactly the same cell. And as the plant grows, it always integrates different environmental conditions. Okay, and my last experimental system is a purely cell culture system where we can completely control what the cells are going to do in the developmental state. So these are inducible pluripotent cells. And here you have you can see these cells here, which are grown in culture in these types of goo, and we can induce them to become specific lignified cells, all synchronously controlling the entire environment of these cells. And we can either genetically or pharmacologically modify them to be able to inhibit their lignin, block specific genes, specific enzymes, or even feed specific genes. To be able to analyze lignin, what we're using is also bulk biochemical analysis. And in my team, what we use is thioacidolysis, which enables us to make thioketals derivatives, which are different depending on their position in the polymer. In that way, we see units with a free C3N compared to internal and units with a free phenolic ends. And you can see that along our different mutants, what we can see is different proportion between internal and terminal residues. And our second method is pyrolysis GCMS, where we thermically break down all the lignin and quantify also after all the different derivatives. And similarly, we can characterize the proportion in our different mutants of one specific unit. And when we compare both our analysis using pyrolysis GCMS and thioacidolysis, we get a pretty good correlation of about 90%. Okay, so all of this is nice, and we wanted to use this entire array to be able to pinpoint and define what is the cellular regulation. So we needed to establish in situ quantitative methods. And the first one that we made, or we tried to optimize from studies of our colleagues before, was to use Raman microspectroscopy. And Raman's microspectroscopy enables you to get different fingerprinting of different cells and tissues within plants. And you can see that depending on where they are, the profile itself and the overall intensity of the profile is completely different. And they can be easily resolved and separated using multivariate analysis. So that was nice. So we decided to take the entire array of all of our mutants and pinpoint on 2 distinct tissues. And you can see here that each time what we're getting is variation in terms of the intensity of the signal between the tissues within each plant and the spectra itself. So taking all this accumulation, what we've done is inputted all of this data into a multivariate analysis to be able to see which were the discriminating band which enabled us to see differences between our mutants in which we had lost specific gene function, thus specific chemical differences in terms of the lignin. And as you can see here, we were able, using this pairwise multivariate analysis and summing up everything together, to see 13 significant Raman band, which were discriminating between the mutants and between tissues. So we therefore created correlation between our bulk biochemical analysis using pyrolysis GCMS, where you can see here on top, where we could quantify a set, the S units, the H units, the aldehyde units, the G units, the total lignin, with the different Raman wave band across all of our different mutants to be able to decipher whether or not we had a linear correlation and thus a semi-quantitative relationship. And we were happy to be able to see that Raman could indeed be used to quantify at the cellular and subcellular level for lignin quantities, and also for S to G ratios. That was quite nice. So we could directly pinpoint to an area of about 200 nanometer square to be able to get a biochemical idea of lignin quantity and composition. However, some units, such as the lignin conifer aldehyde and the aldehyde units, which had been previously suggested to be characterized by the 1625 band, did not correlate linearly. Specifically, in some of our mutants, did not continue the typical trend, which is highlighted here by the dotted line. So other units, also such as the conifer alcohol, could also be precisely quantified directly using Raman microspectroscopy. So that was quite nice. The second method that we used is called the Wiesner test. It was used for a very long time, and people wondered whether or not it could be quantitatively used. And the Wiesner test was established during the middle of the 19th century And it consists of incubating wood with fluoroglycerol and in the presence of acid. And when we look at one of the sections here that we have of our plant, if we incubate it directly with fluoroglycerol and HCl, what we get is specifically a red color of the lignified portions. So we really wanted to identify what it was, and it was previously published in 2002 that the Wiesner test specifically responded to aldehyde of phenylpropanoids, or phenylpropanide cinnamaldehyde residues, and of both having a G, a guaiacyl substitution, and an S substitution. So we tested this in the monomers between our G, S, and H substituted monomers, which were all aldehydes, and you can see here that we had a positive color reaction. However, when we polymerize these units into lignin, into synthetic lignin, making dehydrogenation polymer, we've noticed that the S-aldehyde polymer didn't stain at all. Okay, it was exclusively the coniferal aldehyde polymer. We also saw very interesting positive staining of the coumaraldehyde, but we don't even know if these are lignin monomers because they've not previously been characterized as lignin monomers. So the nice thing is that we were able to decipher between the true target of the Wiesner test staining, which is exclusively coniferaldehyde. So what we've done is to be able to really ensure that there was a chemical substitution, so a chemical linkage of our fluoroglycerol to our synthetic lignin made of one specific monomer. We've compared using size exclusion chromatography our synthetic lignin in the presence or in the absence of fluoroglycerol HCl. And for our coniferaldehyde polymers, you can see in blue when they're not stained, and in orange here when they're stained. And what we're seeing is that they are much more heavier. This is not the case of the synapaldehyde, where we get about the same profiles for size exclusion chromatography. And if we change our carbonyl function from an aldehyde to a carboxylic acid, what we also see is no bridging, okay, and no weighing down. So we were very happy to see that approximately 67 of all the coniferaldehyde residues in our synthetic lignin were actually bound and stained, so covalently associated to a fluoroglycerol molecule. So similarly, we took our total range of mutants in the multiple cell types, like I showed you in the protoxylem, the auxiliary fibers, the interfascicular fibers, and correlated the change in terms of their intensity to the Wiesner test comparatively to their compositional data as a bulk using pyrolysis GCMS to be able to correlate. And you can see here that correlations between the variation of the red color intensity within the different cells and synapaldehyde did not yield a good correlation. Correlation with lignin was not fantastic, but the main correlation that we had, which was very good, with very good efficiency, was coniferaldehyde. And although this technique has been invented in the middle of the 19th century, we find that this is the most reliable and efficient technique to be able to specifically detect and quantify these units in lignin at the subcellular level. So we wanted to compare after with our in situ Raman compared to our in situ quantitative Wiesner test, and correlated changes in Raman intensity of each one of the band along the Raman spectra with the changes of the intensity in multiple cells to be able to see if we have the link. And here I've represented the correlation factors just below as color-coded. Here you've got the scales for both the R and the P values. And you can see that, yes, there was a major peak correlation with the about 1603 centimeter, which is corresponding to all aromatics. So it wasn't really specific. But the two previous bands, which are here dotted, which had been associated to aldehyde, did not actually correlate well at all with the Wiesner test, thereby showing that in terms of coniferaldehyde, the Wiesner test was much more efficient than Raman microspectroscopy. However, this kind of disturbed us because you could see in our initial correlation with the 1625 centimeter -1 band of the Raman, we did have some association with conifer aldehyde. So we decided to be able to compute the differences between this Raman band at 1625 and the Wiesner test, and to decipher whether the position of the conifer aldehyde unit within the lignin polymer would be discriminated by one technique compared to the others. So we therefore took all of our different mutants, quantified everything in different cells, and then correlated these values with the thioacidolysis analysis of the conifer aldehyde position, either as terminal or as internal units. And you're seeing here for the total units that the correlation is much better in gray with the Wiesner test. For the internal units, I mean, we don't have a good correspondence because they just partly represent the conifer aldehyde residues. Whereas for the terminal units, the correlation for the Wiesner test is reduced, but we have a large increase up to almost 70% for the Raman band at 1625. Thereby illustrating that relatively, using the ratio between the 1625 and the Wiesner test intensity, we could get a positional relationship of the conifer aldehyde residue between terminal residue with a free aliphatic tail and internal residues, well, or total residues. Okay, so which is pretty nice because we could similarly at the subcellular level define if the position of the lignin monomers was considered. So we've used all these techniques in a couple of mutants to be able to define how homogeneous is lignin genetically controlled between the different cell types, because each cell type fulfills a different function. So in that case, I focused on 4 different mutations. I've one which is the FAR1, which enables the hydroxylation in meta of YSL into 5-hydroxy-YSL, and the OMT into 5-hydroxy-YSL to a syringeal unit. And the second one are the 2 steps of reduction of the aliphatic tail from a CoA ester to an aldehyde with CCR1, and then from an aldehyde to an alcohol with the double mutant CD4, CD5. And I've always focused all the comparison with 3 cell types which fulfill completely different functions. The support fibers in the intervesicular region, the main conduction through the metaxylem vessels, and the initial conduction of the sap in the protoxylem vessels. So if we compare the overall quantity of lignin, you can see that the different cell types, they have different amount of lignin. Okay, where basically the metaxylem having the most lignin relatively compared to the protoxylem vessel, and even less in the intervesicular. Secondly, if we try to understand how this lignin is being built by the different implication of these genes in terms of their biosynthesis, we see what is the impact of when we remove these genes by reverse genetic engineering. And we see that for some cells, like the protoxylem vessels and the metaxylem vessels, the CCR mutation, this one, doesn't have any impact on the lignin quantity. It only reduces it in the intervesicular fibers. Whereas other mutations, such as the reduction from aldehyde to alcohol, affects only the vessels, well, only the metaxylem vessels, but not the protoxylem. And as you can see, what we're getting is that each time we're genetically modifying one step in terms of the biosynthesis of the lignin monomers, we're differently affecting the lignin quantity in the different cell types. We wanted to know whether this degree of hydroxymethoxylation of the C6 was also genetically controlled differently between cell types, and use the same array of 4 mutants to be able to pinpoint on these 3 different cell types whether they have the same type of regulation. So we compare basically the S to G ratio that we can determine using the Raman microspectroscopical analysis. And we could see that, yes, indeed, the fibers have got much more S than the vessels. But we saw also that the vessels themselves were fairly unaffected by any of the mutations, even the one affecting the S biosynthesis, which was not the case of the intervesicular fibers, thereby showing that the regulation of the composition in terms of the C6 substitution of the residues composing lignin is very specifically regulated for every specific cell. So lastly, we wanted to get everything together for one specific unit, both the aliphatic tail and the aromatic substitution. And using Raman microspectroscopy, we could characterize the proportion of the coniferal alcohol units compared to lignin and compared to the guaiacyl units. So we still basically investigated our 4 different mutations to be able to see how they would affect the content in coniferal alcohol between our 3 different cell types. And you can see yet again that originally in our wild type, in our unmodified plant, what we have is different proportion of the coniferal alcohol, which are differently affected and cell-specifically altered with specific mutations. And even the 2 different vessels, which supposedly do the same function of fat conduction, are differently regulated or differently affected by mutations of the lignin biosynthesis. So we wanted after to be able to go a bit beyond this and define whether or not we had a genetic regulation of the position of specific residues within each specific cell type. And we did this by using the ratio between the 1625 Raman band and the overall quantitative Wiesner staining in purple to be able to discriminate between all units of coniferaldehyde and terminal units of coniferaldehyde in the different mutants that I've previously already showed. And this focusing on our 3 distinct cell types. And we can see here that in between our 3 cell types, what we have is a different proportion of terminal to internal residues, specifically here between the 2 different vessels, whereas the intervesicular fibers and the protoxylem vessel have got the same proportion of terminal to total. We've also seen here that different mutations affect directly the position or the positional proportions of the residue, specifically in every cell type. The protoxylem is pretty resilient to any changes due to this mutation. It doesn't really care. This is not the case for the metaxylem, which can be reduced or increased depending on specific mutations. And same thing for the intervesicular fibers, which can increase or even reduce its accumulation, or reduce here, CD4, CD5, its relative proportions of terminal to internal residues. So this is all good, but we didn't have the developmental context because wood established in a plant grows gradually. So for this, we've used our poplar systems, and in our poplar systems, we also have silenced or genetically reduced the function of key enzymes in the biosynthesis. So here again, we talk about the CCR that I presented, and the C4H is the enzyme responsible of the first para-hydroxylation of the aromatic ring. And as I told you, we can follow the development of the cell types along the developmental gradient of the wood. And here we've got 3 different zones. If we stain them basically with the Wiesner to be able to define the overall amount of coniferaldehyde, you can see for every single cells here for the fibers in F, rays in R and V for vessels, what we have is different relative amount. And this amount changes as the cell age. So we basically have a developmental regulation of lignin monomer amount. We wanted to know whether this was independent of the monomer supply. So can a cell discriminate between monomers, or will it take any monomers that is given to it? So for this, we've used the in vitro systems of our cells, in which we can pharmacologically inhibit lignin monomers or lignin formation. And then we supply externally different lignin monomers. So here, what you have is relative proportions of the fluorescence of lignin. Here, this is when we don't add any monomer, so it doesn't have much fluorescence. And here, what you have is a large increase when we supply these cells with coniferal alcohol. If we change the aliphatic function of coniferal alcohol to ferulic acid or to ethyl ferulate, we see that we've got large reduction. So the cell itself discriminates between the monomers in terms of the amount that it will incorporate. And same thing, we can even alter where the hydroxyl is and shift here between the position in para and meta. So quite nice. It can basically, the cell regulates the type of monomers that it can accumulate in its cell wall. So this was quite important because this showed cell wall layer specificity. And we wanted to investigate this a bit further between the different cell wall layers, in between two neighboring cells. So these are two intervesicular fibers, and from the lumen of one to the lumen of the others, we've got sequential cell wall layers. The cell wall layer separating the two cells is called the compound middle lamella. And then on each side, we have an S1, S2, and S3 layer. And you can see here, using the Wiesner test, we've got different profiles of accumulations of coniferaldehyde, which are interestingly genetically altered depending on the mutations. But this is not homogeneous for every single cell wall layer. Cell wall layers respond differently to the genetic modification for their accumulation of coniferaldehyde, thereby really highlighting that we have a cell wall layer specific regulation. And we could compare all of this and see between all of our multiple mutants what we had in terms of genetic changes, which were not equivalent for every single mutant in every single cell wall layer. So lastly, to put this in terms of the developmental context, right from the beginning, I showed you that the lignified cells were next to each other, okay? And they shared very close organizational presence of one compared to the other. And we wanted to investigate whether this organization was also very important. I'm almost done. And we can see here are 5 different cell types. And what we computed is the effect of genetic mutation on the cell walls in between each one of the cell types for coniferaldehyde using the Wiesner test. And interestingly, what you can see here, encoded with the numbers here and the arrows, is that specific cell either benefited or did not benefit, were kind of reducing the coniferaldehyde accumulation of their neighboring cell. Okay, so saying it's that the entire organization of the xylem itself is regulating cell to cell their lignin composition differently depending on the cells that we're talking. And this is not only due statically, but also changes during wood development, where the interactions of the cells and the contribution of each cell to each other changes with their developmental state. So this is the same thing where we've computed the relationship between the cells, vessels, rays, and fibers in the 3 developmental gradients as the wood ages. Good. So basically, I wanted to give you part of what were the main breakthroughs we made last year. We showed you that we can actually develop tools to do in situ quantitative imaging to image and quantify lignin at the cellular and subcellular level with a resolution of about 200 nanometer square. That the lignin amount in the cell walls are regulated by specific genes in specific cell types. Okay, this is also that the cell wall has itself specific capacity to accumulate lignin, and that the lignin itself is dependent on the interaction and organization of the xylem. And I showed you that the xylem organization changes with species, okay? And it also changes with environment. So overall, what we could define, and this is just a very simplified scheme to be able to show the metabolic flux in the different cell types that I've defined in Arabidopsis for coniferaldehyde in both autonomous and cooperative regulation by cellular neighbors and the impact of each one of the mutations. So here, this is very exciting because you're having these very great regulation for every single cell type with different implication of the neighboring cells, as well as specific genes. So thereby defining that there's probably a specific function of lignin for each cell type, which is not redundant because they don't actually do the same processes. It's not regulated the same way. Good. So I'd like to conclude, and I couldn't have done this alone. I'd like to thank my fantastic research team and my collaborators, as well as the funders, and yes, and the people we're collaborating over in Japan and at Stockholm University. Thank you.